Hi, Shunlei.
Hey, Carlos.
Are you okay?
Oh, yeah, good. Yeah,
Great. Thank you so much.
sure.
Well, it's good morning for you. It's correct? What
What
time
is
is it there?
What did you say?
What time is it
8
for you?
o'clock in the morning.
Okay, there you go. So, the microphone is open.
Sì,
Voilà.
infatti la parola di microfono.
Here. Okay.
Fa un po' come
Well, Professor Shunlei, we are waiting for other members of the committee.
Okay.
Yeah.
All right. No problem.
Well, I don't know if it was clear. This is a public exam. Okay, just to clarify that. Okay. So actually, here we have some people here in the room.
Okay.
Okay. They are just watching the
Oh, I see.
presentation of Fábio. We have some person here, some is coming here in Brazil. That I hope that we finish in less than 3 hours, I think.
Yeah.
But so, Julie, I would like to ask you: did you receive my email? About the image that we would like to process.
Yeah.
Okay.
Let me check.
It's around 50 pages. Actually, 50 subjects. Not necessarily.
Oh, 50 post-mortem brain images. Is that?
Where is the microphone? It's in your computer.
Yeah.
Okay. No, it's not. You know that we have an external microphone here. It's possible to use, but it's working now, so it's fine.
Yeah.
Hello.
Hello.
Well, good afternoon. Hi. It's my pleasure, Professor Maria, and Professor Christian. My name is Carlos Garrido. Actually, Professor Chulei know me because he visited us 6 months ago, I think. Okay. Well, I think that we can start just to, okay. Well, good afternoon to the majority and good morning for Professor Chulei. Today we have the exam of the PhD student Fábio Otsuka. It is a public exam. In fact, we have some people here. I don't know if you can see here that we have some people in this room here. It's a public exam. So it's open for everyone to watch. And maybe we have some people on internet, okay, because part of this exam is transmitted by YouTube. Okay. Well, Fabio will defend his work entitled "Quantification of Paramagnetic Human Ions in Human Brain Tissue: Correlation to Quantitative Magnetic Resonance Imaging," in order to obtain the title of Doctor in Science in our graduate program of Physics Applied in Medicine and Biology of the University of Sao Paulo, Brazil. Firstly, of course, I would like to thank the presence of everyone here, but especially the committee members. All of them are online. They kindly gave up their time and experience to evaluate Fabio's work. The members of this very qualified committee are Maria Fittipaldi, Associate Professor at the University of Florence, Italy. Dr. Fittipaldi is the chief of the most power and electroparamagnetic resonance laboratories at the Department of Physics and Astronomy in the University of Florence. Christian Langhammer, Associate Professor at the Medical University of Graz in Austria. Dr. Langhammer has worked for over 12 years in quantitative susceptibility mapping and postmortem MRI. And Professor Chun-Lei Liu, full professor in the Department of Electrical Engineering and Computer Science and the Helen Wills Neuroscience Institute at the University of California in Berkeley, USA. He is the co-director of Henry Weller Jr. Brain Imaging Center. Dr. Liu is MRI specialist with focus in diffusion imaging and quantitative susceptibility, and in the last 10 years have been working with some biophysical models. I myself, Carlos Garrido, Associate Professor in the Department of Physics at the University of São Paulo, Ribeirão Preto campus. Thank you, Professor, for your time. This exam has 3 parts. During the first part, the candidate has no more than 60 minutes for an oral exposition of his work. It's okay, Fábio?
Yeah.
60 minutes. In the second part, maximum time, okay? Between 40 and 60 minutes. In the second part, each examiner can discuss with the candidate about the work or a presentation in hand or tested documents that he presented before to the members of the committee. And the last part is a secret section among the committee members for the final result. This final result is made public at the end of the exam. It is important to say, because it's important to say for you that in our university, a small modifications are allowed in the submitted document to the committee after the exam. At the end of the exam, it's possible to suggest some details to add to the document if you think so. Okay, because we know that it's important. The most important thing is a very good exam, a final document. Please, if you have any doubt. No doubt? Okay, well, we can start. Fábio, please.
Yeah.
I suggest to ask first if it's everyone.
Yeah. So, can everyone see my slides, my presentation? Yeah. I think so.
Yes, yeah, yes.
Okay. So first of all, I'd like to thank everyone joining my presentation here, all the members of my committee and everyone here in this room. So I will present my thesis entitled Quantification of Paramagnetic Ions in Human Brain Tissue, a Correlation with Quantitative MRI. Okay. So I structured this presentation in a similar way I did with my manuscript. So I divided all the results into 3 parts. So the first part will deal with all the imaging aspects of this work. The second part will deal with spectroscopic aspects of this work. And the third part will be merging all the results from both imaging and spectroscopic in order to gain further insights of the contrast sources of QMRI. Okay. So we will start with a brief introduction. So we have iron in the brain. Iron is probably the most abundant metallic trace element in the brain. So it participates in a lot of different biological functions in our body. such as oxygen transport, enzymatic reactions. It is is a cofactor of many metalloproteins, such as ferritin, transferrin, and others. And specifically in the brain, it also participates in the myelin production and also in neurotransmitters metabolism. So iron is a very important metal in our body. Yeah. And some studies have observed that iron concentration increases over aging. So here in this graph, you can see the iron concentration in the y-axis and the age in the x-axis. So we observed some increase of iron concentration on different cerebral regions. Yes, so this I must say that this model was taken from this study by Hago and Surrender. So we can see that at some point, iron stops accumulating in the brain. So we can establish like a range of 30 to 90 years in which iron stops accumulating in the brain, depending on the region. But what's interesting is that some differences in the iron concentration in the brain was also observed on some neurodegenerative diseases. So, for example, we have Parkinson's disease, where we have increased iron concentration in the substantia nigra compared to healthy controls. Yeah. So it's obvious that iron has a significant participation in the development of neurodegeneration in the brain. But the problem is that all of these studies only worked with ex situ tissue, so they worked only on post-mortem subjects. And well, of course, we have many different techniques in order to quantify these iron concentrations. So, for example, in this study, they used colorimetry, but we also can use mass spectrometry or many other techniques. But the problem is that if we want to know exactly the role of iron over the progression of these diseases, it is interesting to have an in vivo method in order to quantify or at least evaluate this iron distribution of the brain. Yeah. So here's an overview of what I said before. So when we have iron balance in the brain, in the healthy brain, it is responsible for many important processes in the metabolism, myelination, neurotransmitter, oxygen transport. But when we have iron overload, it is linked to neurodegeneration by means of processes like neuroinflammation, oxidative stress, protein aggregation. So, but the link between these three things, so iron overload, these processes here, and the neurodegeneration is not very clear, and that is because we don't have means to evaluate the iron content for in vivo conditions. So MRI is a promising tool in order to evaluate this iron distribution in the brain because it allows us to indirectly quantify magnetic susceptibility effects in the brain. So, for example, we can perform an R2 star image or a QSM, which is related to magnetic susceptibility. And, well, there are some studies that have performed some validation of these techniques on high iron content regions, and they showed that actually we have a correlation between qMRI and iron concentration on these high iron content regions. So here in this image, I'm showing also a histochemical technique that stains for iron. So darker blue means that we have higher iron concentration. And well, when we compare with MRI, we can see some kind of agreement, mainly in the basal ganglia, that is this central region here. So we can see some kind of agreement, which indicates that at least for these regions, MRI is able to quantify our iron concentration in the brain. So the problem of the validation I'm illustrating here. So of course we have images, so quantitative images at post-mortem conditions, right? So QSM and RT-star. And well, as a first step of validation, we have to look at different possible metallic sources that can contribute to these positive magnetic susceptibility. So, well, we have to quantify iron, copper, and also other metals, zinc, manganese. But another question that we can also do is most related to the second step validation, is more related to specific molecular forms of these metals. So, for example, here I'm showing for iron, we have different sources of iron in the brain. So we have labile iron pool, LIP, or neuromelanin complexes. The ferritin core is also rich in iron. And for copper, we also have different proteins related to these metals, such as ceruloplasmin and superoxide dismutase. So the question here in the second step validation is, amongst all of these molecular forms, which one is mostly related to the QMRI, right? So in order to do this, for images, we have to work with post-mortem data because, of course, we have to later on do some quantification on the tissue, so that is only feasible on post-mortem condition. So, but one problem about post-mortem is that we can perform this either on in situ condition or ex situ condition, and later we will see that these may have some relevant differences between them. For the first step, we chose to use the inductive coupled plasma mass spectrometry, SPMS, because it has a high sensitivity for trace elements, and also we can simultaneously quantify these different elements in the sample. And for the second step validation, we chose to use the electron paramagnetic resonance, EPR, because this technique is very sensitive to paramagnetic ions in the sample, but it also is very sensitive to many different magnetic interactions that can occur within the paramagnetic ions. So it can differentiate the same ion. For example, the iron 3+, it can be differentiated according to its chemical environment. So it is a way in which we can determine different molecular forms of this paramagnetic ion. Okay. So there are some questions of diseases that I'm going to present here. So the first two questions actually are related to the QMRI. So the first one is R2 star and QSM, are they both equivalent in terms of iron mapping throughout the whole brain, or do they have some differences, right? And also about the in situ and ex situ conditions, how much they differ in terms of desensibility. As for the spectroscopic part, we can ask, how are these metals and how are the paramagnetic ions distributed in the brain? So are they heterogeneously distributed? Do we have differences among subjects or regions? Yeah. And also, if the both techniques that we are using, the SPMS and EPR, are they correlated in some way? Yeah. And finally, when we merge all this data together, will we observe some kind of correlation between QMRI and spectroscopic measurements, which would indicate possible contrast sources of QMRI? Yeah. So, of course, we have some hypotheses for these. So for both images, we expect to observe some kind of similarities in regions where we have stronger magnetic susceptibility contributions. But for regions where we have low magnetic susceptibility contributions, we expect some kind of discrepancies because R2 star isn't can also have other contributions besides the magnetic susceptibility. Yeah. As for in situ and ex situ conditions, we also expect to observe some differences because we have, for example, the oxygenated blood in situ conditions, which is totally removed in ex situ. We also have air bubbles, which can contribute to the signal. So we expect to observe differences in these as well. Based on literature, we also expect the paramagnetic ions and the metals to be heterogeneous throughout the brain. Yes, so we expect to observe some agreements to the literature. And we also expect a correlation between SPMS and EPR. That is because although EPR quantifies only the paramagnetic ions related to these metals, essentially they are looking at mostly probably at the same element. And as for the combination of all these data, we again agreeing to the literature, we also expect to observe some kind of correlation when we look to iron-rich structures. And as for EPR, that is not in the literature, we also expect to observe some kind of correlation, especially for the ferritin signal, which I will be showing later on in this presentation. So moving on to the first part of our results. Yeah. So here's the full workflow of this work. So we have at the Clinics Hospital in São Paulo, we have the subjects inclusion to this study and the image acquisition and the sample acquisition, which will then be, the images will be processed. Actually, they were processed here in Ribeirão Preto, and the samples were acquired as well. And so for the images, we did the image processing, post-processing analysis, the sample, almost the same thing. We did the spectroscopic measurements and analysis, and then we combined both data in the third part of this presentation. So for the first part, we are dealing only with this step here of the workflow. Okay, so for the image acquisition, we did with 15 postmortem subjects in situ imaging, and amongst these 15 postmortem subjects, we also imaged 2 postmortem subjects on ex situ conditions. We did it with a 7 Tesla MRI scanner with a gradient echo, 3D gradient echo sequence. So here are the parameters of the sequence we used. With the magnitude images, we did R2 star mapping by using a mono exponential fitting. And for the phase images, we processed them for the QSM using the MEDI toolbox. So the choice of the MEDI toolbox was actually because it is a widely distributed tool for QSM processing in the literature. It is a very used technique. And we also had a greater familiarity with the code of this in comparison to other techniques. So that's why we chose to use the MEDI. For image segmentation, we performed it on a visual basis compared to brain sections during the sample acquisition. So we compared the images to the coronal sections of the brain and manually segmented the structures. So all the structures are listed here that we segmented on the images, which is a globus pallidus putamen called the nucleus, hippocampus, entorhinal cortex, precentral cortex, red nucleus, and substantia nigra. For each ROI on each site, we calculate the mean R2 star and chi values, and then we perform the correlation test and linear regression between these values. So all these statistical analysis we performed on a global basis, which means that we included all the regions for the analysis, but we also performed an ROI level analysis, which was this analysis individually for each ROI. So the first thing that we are looking here is the comparison between in situ and ex situ conditions. So the first thing that we can notice is a decrease in both R2 star and magnetic susceptibility when we move to ex situ condition. Yeah. When we're looking at in situ conditions, we can also observe strong signal arising from the oxygenated blood. So in the red arrow, I'm showing one example, but we can also observe throughout the whole brain. And that contribution seems to vanish when we move to ex situ, which is expected, since for ex situ condition, all the blood should be removed, right? But on ex situ condition, we have another problem, which is air bubbles. So in yellow, I'm showing one example of an air bubble, but we can also observe on the circles of the brain. And this is a huge problem because air tissue interface contains strong paramagnetic gradients, which affects strongly the QSM map. So in the QSM, we can see lots of different artifacts arising from these air bubbles, which are these X crosses that are spread out throughout the whole image here. Yeah. For a quantitative analysis, when we compare all the ROI data, we can see a good correlation between ex situ and in situ condition for both R2 star and QSM. However, we observed that the ex situ conditions, we have a decrease in the values, which was already observed for qualitative analysis. Yeah. And this indicates that overall the contrast is maintained when we move to ex situ conditions. But in order to evaluate these individually for each ROI, we will need more subjects. So remember, we only have 2 subjects for these tests. So it is an initial study. However, we also observed a similar result in a previous study from our lab, from a previous PhD student that worked on a similar way that I did here. I also calculated a percentile decrease between in situ and ex situ conditions. And what we can see is that QSM seems to be more sensitive to these changes in ex situ conditions, because the percentual decrease can go up to 150%, while the R2 star is within the range of 20 and 50% of decrease. Okay, but interestingly, in regions of high iron content, the basal ganglia, we have lower percentual decrease on QSM, so it is comparable to R2 star. So this indicates at least that for these regions, at least we can expect both techniques to perform in a similar way and be less affected by these differences between conditions. Yeah. Okay, so now comparing both maps, the QSM and R2 star maps on in situ conditions. In our global level analysis, we observed a strong correlation between the data. And the linear regression results, well, it disagrees with the literature, most parts. So possible sources for this disagreement are the R2 star dependence on the magnetic field strength. So it has been observed that R2 star are dependent on the magnetic field strength. So it increases with magnetic field. So that's one possible source of disagreement for these studies. Another thing is that in vivo condition may be different from post-mortem condition, but that needs still to be evaluated further. And also the imaging processing pipeline, which can also interfere with both QSM and R2 star results here. But although the slope deviates from the literature, the strong correlation is agreeing to the literature, which indicates that both techniques, at least globally speaking, are correlated. When we do this individually for each ROI, we can observe structures which presented a strong correlation between techniques, which are mainly iron-rich structures. But we also observe regions which were partially or unilaterally correlated, which was the hippocampus and substantia nigra, and also regions which did not show any correlation at all. So this suggests that the agreement of QSM and R2-star are region dependent. So what we observe here, for example, is that iron-rich structures showed a good correlation. So these structures, they present strong magnetic susceptibility contributions. And that's why we observed a strong agreement between them, while other regions which have lower iron content, they do not show a strong correlation. But interestingly, in the substantia nigra, we know that it also contains high iron concentration, but it also did not show any strong correlation. So that's an interesting result. This possibly the neuromelanin could be affecting the contrast in this region, but we have to look further into this. So in conclusion, for this imaging part, we observe a strong correlation between R2-star and QSM at a global level analysis, and only at an ROI level analysis only for iron-rich structures, with the exception of substantia nigra. As for in situ and ex situ comparison, our initial study showed that both R2-star and QSM were decreased at ex situ conditions, but at a global level, they were correlated. The in situ and ex situ were correlated, which indicates that the main contrast is still maintained. And also that QSM seems to be more sensitive to these changes, right? So the changes to in situ to ex situ conditions. Okay, so moving to the second part of this presentation. So now we are here. For the sample preparation, we did the sample extraction on the same 15 post-mortem subjects we imaged before. The sample extraction was performed by an expert neurologist. The samples were separated. Then we measured the wet mass of each region. The samples were then lyophilized, and the dry mass was also measured. Then the dried samples were grinded to powder in order to do the spectroscopic measurements. Okay. So the EPR measurement was performed on an X-band spectrometer. So the parameters are indicated here. For each spectrum, the empty tube signal was also removed in order to remove any background contributions. And the mass of each sample that were measured was also obtained in order to normalize the spectrum by the dry mass of each sample. As for the processing of the spectrum, they were performed basically using the Easy Spin toolbox. As a pre-processing step, we did a baseline correction, which was a standard approach for narrow features. And as for the broad feature, we did a dedicated pipeline for analysis, which was mainly developed during my internship at Leiden University. Okay, so moving on to the results. Here I'm showing you the wet to dry mass ratio on the different regions. So red dots indicate values in the literature as a comparison. So we can see that overall we have an agreement to the literature, but the global pallidus and the red nucleus showed slightly higher values than was reported in the literature. Okay. The white to dress mass ratio is related to the water fraction of each tissue. So what we can see is that red nucleus presents the lower water fraction compared to other structures, while higher water fraction can be found in the caudate nucleus, putamen, hippocampus, and entorhinal cortex. Here. Another thing that is interesting is that the locus coeruleus and the substantia nigra presented similar water fractions, which is expected since both of them are higher, contain higher concentrations of neuromelanin, and that could potentially explain why they have a similar contrast on neuromelanin sensitive images. As for the APR, we observed similarities between the APR spectrum of different regions and between different subjects. So overall, they all presented the same 3 peaks, paramagnetic peaks, which were I'm indicating here as a G equals 4.3 signal related to iron, copper peaks, and also a broad feature located at G equals 2.01. So most all of them presented these signals, but I should say that there were some subjects, some regions that presented additional peaks, and we chose to not include them into our analysis here. Another thing that it is interesting is that the broad feature that we observed is very similar to the ferritin, to the horse-spilling ferritin signal. So all the processing of this broad feature was performed considering a ferritin model of the ferritin model. So here I'm showing you the fitting. On the left is the horse-spilling ferritin fitting, and on the right is the human brain sample fitting. So we can see a good agreement between the fitting and the experimental data. Yeah, so when we look individually at each peak, we can see also a good agreement between. So the G equals 4.3 signal is related to high-spin iron in a rhombic symmetry. It is commonly found in many biological systems, and specifically for the brain has been attributed to the labial iron pool signal, LIP. But it also is observed on ferritin samples. So we hypothesize it to be the iron at the ferritin shell instead of the iron at the core of the ferritins. In a previous study that we published in 2021, we also observed paramagnetic behavior with temperature of this signal. As for the copper, well, it has a very known shape. So it presents an axial symmetry. So we have 2 components for its modeling. Possible sources can be ceruloplasmin or the superoxide dismutase, but further studies need to be performed. And in our study, we also observed the paramagnetic behavior with the temperature of this peak. As for the broad feature, as I said before, it presented a similar shape to the ferritin spectrum, but the ferritin spectrum fitted very well to the data. In our study, we also observed an antiferromagnetic behavior with the temperature, which was also observed on pure ferritin signal. So all of this suggests that this broad feature that we are looking here is indeed related to the ferritin signal, and therefore we're going to be treated as a ferritin signal from now on. When we look at the fitted data, so here in this table, I'm showing all the parameters that were fitted for each paramagnetic ion investigated in this study. So A and B here indicates the model of the ferritin, because the ferritin was modeled as a superposition of two different systems, both located at and fixed at 2.01, and then with different zero field splitting parameter between them. So what we can see is that these parameters, they in parentheses are indicated the standard deviation. So we can see that overall the standard deviation of these parameters are low, are very low, which indicates that there is not much difference of these parameters when we amongst regions and subjects, indicating that different regions and different subjects present similar paramagnetic ions at similar environments, which indicates that these paramagnetic ions are indicated to the same molecular sources in the brain. Okay, so now moving on to the quantification of this data. So firstly, I'm showing you the ICP-MS results here. Dashed lines represent a range found in the literature. So the range of values found in the literatures. And here I'm showing you a subject-wise distribution. So each one of these values here represents one subject. Okay. So what we can see is that overall we observe some differences among subjects, especially in the zinc and the copper. And also that zinc, the manganese and aluminum that we quantified, they deviated from the literature values. But the iron and the copper are mainly within the range of reported values. When we do this at ROI wise, so now each one of these represents one region, we can see that the iron is mainly lower. Our quantified iron is lower than what has been reported in the literature, and the zinc as well is lower and reported to the literature. But what is interesting is that the basal ganglia, which are these regions here, presented increased iron content compared to other regions. As for the APR, when we analyze these, we can observe similar behavior between the ferritin, the broad feature to the iron from ICP-MS, and the copper ions to the copper. And this happens both at the subject level and ROI level as well. So here's the broad feature, here's the iron, and here's the copper ions, and here's copper. So from these, we expect to observe some correlations, and that's what we did. So when we performed a correlation test, we observed a strong correlation between the broad feature and iron and the copper ions and total copper concentrations. So this is a good indication that indeed this broad feature is related to the ferritin, in addition to what I said before as well. So this is a good indication that this is the ferritin signal. While this correlation here with copper was already expected, since, well, both of them are representative of the copper. But what this also tells us is that this copper ion that we are observing on EPR correspond to the majority of copper present in the sample due to this strong correlation. But it does not correspond to all of the copper in the sample. So that's one thing that we have to keep in mind, is that EPR does not account for all the metal in the tissue. Okay, so there are some forms of iron and copper that we cannot quantify via EPR. So in conclusion, we observed heterogeneous distribution of both ICP-MS and EPR data. This was both among subjects and amongst regions. As for the EPR data, we observed that the fitting pipeline that we developed showed good performance, so it fitted very well to the data. And the spectral composition of of EPR spectra showed similar behavior between subjects and regions, which indicates that the quantified paramagnetic ions are the same molecular sources between regions and subjects. We also observed good correlation between SPMS and EPR, especially the broad feature and the iron, which indicates that the broad feature is related to the ferritin, of course, in addition to the other findings. And also we observed a good correlation between the copper ions and copper. And again, EPR does not account for all the molecular forms. So if we want to quantify these other forms, we have to use other techniques in order to complement our results. So now we move to the last part of our results, which we will be merging both imaging and spectroscopic data. So now we are here. And for these, what we did, we created 4 general linear models using QMRI as independent variables and both ICP-MS and EPR as QMRI as dependent variables and ICP-MS and EPR as independent variables in order to try to explain the contrast of QMRI based on ICP-MS and based on EPR. From the GLM results, we performed the correlation test and linear regression only for the elements which showed statistically significant results here, which I'm highlighting in the dashed red lines here. So basically it was the iron and the ferritin signal. Yeah, we did. And these tests was performed at a global level analysis again and at an ROI level analysis. Okay, so for the global level analysis between R2 star and ICP-MS, we can observe a strong correlation between R2 star and iron. And on the left, I am also showing only considering the basal ganglia regions structures, which is a comparison to the literature. So what we observed is that, it agrees to the literature because the literature also found a strong correlation between these variables. But when we compare our slope to the literature values, we observed some differences between them. So the values here that I'm showing you are already corrected for the R2 star variations with magnetic field. So the differences here are due to other factors. There were some studies that observed that R2 star differences due to different centers are very low, so they are not sufficient to explain all these differences. So what possible sources for this disagreement could be the number of subjects used in each study, the imaging processing pipeline could also have affected the results. When we look at comparing the QSM to ICP-MS, again, we found a strong correlation, which also agrees to literature. And our slope, 0.79, is more in agreement to the literature. It also deviates a little bit, but it agrees more than what happened with R2 star. Okay, these differences, again, could be explained by different image and processing pipelines between what I used here and what the literature has used, and also differences in conditions. Okay, so in situ, ex situ, frozen, fixed brain tissue, these all could have influenced the results. Comparing to theoretical predictions, which is 1.32. We found a strong deviation. And also compared to experimental ferritin data, we also observed a strong difference to this. So, but this was actually expected because theoretical prediction considers that the only contributor for the contrast is actually fully nucleated ferritin, which is not the case in the brain sample. So we have other iron sources there that can also be contributing. So yeah, that was an expected result here. When we compare to the APR, we also observed a strong correlation to the ferritin signal at our global level analysis. And it kind of it corroborates the hypothesis that amongst all the iron in the brain, the ferritin is the main contributor for the contrast. And this was also observed for QSM. So we also observed a strong correlation to the QSM. I did not do any analysis of the slope here because EPR, as it is now, is not an absolute quantification method. So we need to, in order to extract absolute quantification, we need to have a standard sample in order to convert the quantification. So therefore, the slope analysis was not performed here. And finally, I think that this is the most important results of our study because, as we know, the literature does not have any of these kind of results, which is the ROI analysis of these comparisons. So we did these correlation tests individually for each ROI. Okay, so what we observed here is that we can observe differences in correlation according to the structure in which we are looking. And based on this, we preferred to divide them into 4 classes of structure based on this correlation. Yeah. So the first class here presents a strong correlation, which we consider R squared higher than 0.5, and it strongly correlated to both iron and the ferritin signal on both R2 and QSM maps. Yeah. So essentially, these regions were the basoganglia regions with also as well the red nucleus. And what they show us is that on these structures, iron is the main contributor for the contrast, which based on the literature was already expected, right? So this was an expected result. So for the second class, we observed a weak and unilateral correlation, so it was lower than 0.2. It was only the entorhinal cortex. What we can say here is that iron and ferritin seems to contribute to have low contribution to QSM, but apparently enough to generate some kind of correlation here. But for R2 star, we did not observe any correlation. So other mechanisms are contributing to the R2 star value. The third class is the substantia nigra, and this is an interesting result because it's partially correlated to the total iron concentration, but did not correlate to the ferritin signal. So what this indicates is that although iron seems to be a strong contributor for the contrast, it is not from the ferritin. So it should be from other iron source in the substantia nigra, possibly the neuromelanin, but we need to further investigate this. And for the fourth class, we did not observe any correlation. So this indicates that neither iron or the ferritin are sufficient for the contrast of these structures. From our data of ex situ in situ, we could also perform an initial analysis of how these conditions are, how sensitive are these conditions to iron and ferritin. So what we observed is that we have a decrease in the correlation on the ex situ condition. And also the slope is lower, which indicates that the ex situ condition is less sensitive to iron and also to EPR compared to in situ condition. But again, this is an initial result. Okay, so in conclusion, we observed a global correlation between iron and ferritin to quantitative MRI, which is in agreement to the literature. Some of our results of the slope of the linear regression were different from the theoretical predictions and different from the literature. And we also observed some differences between in situ and ex situ conditions. So what we believe is that the oxygenated blood could be contributing for the in situ signal. Yeah. As for the ROI level analysis, we had some interesting results here. So what we observed is that both iron and ferritin have different contributions according to the region in which we are looking. So we were able to divide 4 classes of structure. And comparing ex situ to in situ data, we observed that in situ seems to be more sensitive to iron and ferritin. But again, we only did it for 2 subjects. So in order to have more confidence into our results here, we need to include more subjects for this. Yeah. So in order to conclude everything and point for future perspectives, firstly, I'm going to answer all those 5 questions that I presented before in the introduction. So concerning the first question, yes, ArchoStar and QSM seems to have equivalent sensibility, but only for iron-rich structures, which were the first class that I presented before, and the second class as well. But for other structures with lower iron concentration, they presented different contrast mechanisms. For in situ and ex situ conditions, our initial results show that both of them are correlated, so the overall contrast is still maintained between them. Concerning the metal and paramagnetic ions, we observed the heterogeneous distribution of them. Higher values of both iron and ferritin were found in the basal ganglia and the red nucleus as well, whereas the copper and iron copper was found on red nucleus, substantia nigra, and locus coeruleus structures. ICP-MS and EPR were strongly correlated only for iron and the ferritin signal and copper ions. And when we compare both imaging and spectroscopic data, we found an agreement to the literature, which is the QMRI correlated to iron. But we also found it correlated to the ferritin signal. And again, on the ROI level analysis, we found the differences in the correlation according to the structure, which we divided into 4 classes of structures. Of course, there were some limitations of this study that I should be mentioning here. So the first one is the QSM and ArchoStar processing algorithms, which were limited by the acquisition parameters that we acquired. So, for example, we cannot perform multi-exponential fitting of the ArchoStar data, and other more complex processing of QSM were not also suitable for these images. And another thing that we have to look on is the agreement between the segmentation on the image and the actual sample of the tissue. So that's one thing that we still have to work in order to improve this agreement. Okay. As for the APR, the main focus of this project was the broad feature or the ferritin signal, but we also have other paramagnetic ions that we were able to quantify, and they should not be discarded from the analysis. So we have to include the analysis of these. But one problem that I did not say during this presentation is the possibility of iron oxidation processes during the sample handling. Yeah, so the iron oxidation could increase the iron 3 plus concentration in the sample. However, we expect it to contribute mostly to the G equals 4.3, the labile iron pool in the sample, and not to the ferritin, because the ferritin core is a very stable complex. So we do not expect iron to freely enter and exit this core. So, but yeah, that's one limitation of the study. And as for the ex situ images, we only did for 2 subjects. Again, we need to increase the number of subjects. But before we do this, I believe that we need to optimize all the pipeline for the ex situ images, because it is not easy to do. It is very difficult to do this in all steps for the images, since the preparation of the brain until the processing of the brain. So for future perspectives, based on these limitations, we can expect to include other MRI techniques, such as the multi-exponential modeling of R2 star, and also the separation of components of QSM. So here we try to do the decomposed QSM on one subject that we have here. So I'm presenting the results here. But of course, the imaging parameters that we used are not optimized for this kind of processing, but it seems that it is a feasible technique that we can expect to include here. Yeah. Neuromelanin sensitive MRI is also a good option, since we also observed that strange behavior of substantia nigra with iron and ferritin. So yeah, so we can expect to include this. And also one thing that I did not mention throughout this presentation is about the locus coeruleus, but that is because it is from our images, the locus coeruleus was invisible, so we could not segment it on the images that we have. And the improvement of workflow, as I said, in order to agree the imaging and the brain. So what we try to do is to develop this device in order to perform a brain slicing which agrees better to the images. Yeah, so essentially we do the ex situ images. With these images, I create this device, then we print it, we place the brain, and then we can slice it exactly at the same slices on the images. So we hope that this should help for a better agreement of ROI segmentation and sample excision. For APR data, we expect to look better at the copper peak. So we have to optimize the acquisition of this region and investigate better which molecules could be related to the copper, because copper also presents very important roles in the brain. The effect of brain oxidation is more difficult to analyze, but if we think in using like fresh fixed sample or other methodologies in order to avoid oxidation, we can better investigate its effect on EPR data. Yeah. And finally, if we include more subjects in the ex situ part, we can improve the results, our initial results from these theses. And we can also think about trying to do this in a disease group. So we will have the same results in Alzheimer's or Parkinson's disease case or not yet. And also to include other histological evaluations in order to investigate how everything is connected, right? So metal, paramagnetic ions concentration, imaging, the proteins related to these diseases, how everything is connected. Yeah. So I like to thank, firstly, the physics department here for allowing me to perform this study, my colleagues at the Clinics Hospital in São Paulo, and my colleagues at Physics Institute and Leiden University, which helped me a lot throughout these experiments. And also for the Professor Jabbar and Professor Bafa for allowing me to perform the EPR measurements here, Professor Otsuka as well for its contribution for EPR, Maria Otadui in São Paulo for all the help in acquiring and obtaining the subjects, the images, and the samples, and Jian as well, which was a previous study that initiated this study and guided me through my first steps here. The InBrain lab, which is my lab, and all the colleagues here, and as well the funding agencies, which helped me here. Yeah.
Wow.
So that's the end.
But thank you, Fábio, for the oral presentation. So I do move to the second part of this exam. For this, I would like to invite Professor Fittipaldi to do her considerations about Fábio's work.
Okay, so thank you. First of all, I want to congratulate with Fábio. Well, you have done a lot of work. You have reported a great amount of data and also used the different techniques and also have rationalized the data and correlate them. And as I said, there is a lot of work behind, so congratulations for it. And also the way the thesis is written is clear, direct to the point, so I also appreciated that. And also your presentation today was very good, so thank you. So I don't know, is this also the time for the question, I guess, right?
Yeah.
Yeah. Okay. So I have few questions, and some of them you have somehow solved during the presentation, because actually, for example, I didn't find the experimental parameters that you have used to acquire your EPR data. And now I discovered that maybe I did it wrong, but I didn't find, for example, the modulation amplitude, and I have seen now that you have used 5 millitesla of modulation amplitude to acquire DPR data. Is this correct?
Yeah.
Okay. So I think it's quite high value. And this could be also the reason why maybe you lose some of the copper signal. I mean, if you have a species somehow in which for which this value is too big, then maybe the signal, you do not reveal the signal. And also I had the question: why did you restrict yourself to do the measurement, EPR measurement at room temperature?
it's mostly because we had during my project we had some problems with the temperature controller of our EPR, so that was a problem. But what we did was actually measure at other facility, which was about these results here, but it is a very limited measurement. So we only did for 3 samples of temperatures. Okay, so now you see. Yeah, so it was only for these 3 samples here, which was performed at other facility. So for these samples, we were able to go from 5 Kelvin up to the room temperature. But now we fixed the temperature controller, so we expect to be able to run more variable temperature measurements.
All right. And I have also a question about this figure that you have reported, the absorption versus temperature. Do you report also some fit of this data
Yeah.
for the different region? Yes, I would like, I don't know if you remember, but so the absorption is related to the quantity that you have, right?
Yeah.
But the behavior with time, is this also dependent on the region or not? I mean, what I mean is that, of course, you should make a reference to the absorption that you see, let's say, at very low temperature. And I would like to know if the decay of the signal with the temperature has the same behavior for the different region or not.
Okay. So actually, not exactly the same. We observed some differences. So yeah, what I'm showing here is different temperatures. Of course, since they have different concentrations, they will present different values here. But when we move to the broad feature, actually what we observed is some small differences. So basically what we found is differences at the blocking temperature, which seemed to be different between the globus pallidus and the substantia nigra. But since it was an initial result comparing only two samples, I can't say for sure if this would translate to a general case. So, but what we are planning to do now is to perform more measurements like this. So what we have done recently is we are collaborating with Leiden Institute, Professor Martina. So we sent her some actually, I think we did not send yet, but we are planning to send some samples for her to do these measurements. So I think we will have more data about this.
Mm-hmm. Okay, thank you. Yeah. It is possible also, I think, that some of the copper that you lose depends on the temperature at which you do the experiment. It could be that at lower temperature you could reveal other species.
Yeah, exactly. I totally agree with that. But since our main focus was the broad feature, we did not thought very much about trying to optimize the signal of these other species. But yeah, in the future, when we look, when when we look at the copper signal, we should be optimizing these parameters.
All right. I would like to understand a little better. For the R2 star that you have measured, it is important to know also how the transition metal are distributed in the tissue. Is this true or not? Or is it only related to the concentration and not really how it is distributed?
Yeah, that's a good question, actually. I think that it depends on both of them, I would say, because if we don't have enough concentration of a metal, it will not produce sufficient magnetic susceptibility effects, so it will not affect the signal. But the contrast, which is a difference of signals between different regions of the images, are mostly, I would say, related to the distribution, because if we have the same distribution of the metal throughout the whole brain, then of course it will not produce different signals, and we cannot differentiate in the images.
Yes, I don't know if this is somehow has been included, because, for example, when you have this the iron signal from the core of the ferritin, then you have a lot of iron that is in one protein.
Yes.
And while, for example, let's say the iron 3 plus, which gives you this signal at 4.3, then it's an isolated iron. So is this, when you correlate, let's say, the R2 star data with other measurement, are these issues taken into account?
Yeah, they were taken into account.
All right. Yeah, it's a pity that somehow you could not correlate what you have observed with the disease, the eventual disease of the people, so that it could give you some more direct information. But I think that you have set a good way to continue your work. Yes. About the manganese, so you say that the manganese ion is also in lower concentration with respect to copper and so on, and you have disregarded because of that.
Yeah, actually, what we did, we actually included manganese into the model. So, but what we observed that it was not enough. Statistically speaking, it was not enough to explain the contrast on both images. So that's why we excluded from the correlation and linear regression tests.
All right. But it could also be, for example, I think probably relevant for the point of view of disease, and therefore, although not relevant for this analysis, it could be useful to detect it with EPR. I think it should be detectable with EPR. What is the oxidation state of the manganese?
Yeah, so we did not focus on manganese, and from what I remember of our spectra, we also did not observe a manganese signal, but that could be probably because it is hidden in this broad feature, the signal to noise ratio was not enough to observe any manganese signal here.
Or the modulation amplitude is too big because the signal of manganese should be quite sharp.
Yeah.
I think. Okay, thank you. I think I'm satisfied with the question, so thank you. And
Thank you.
Thank you, Professor Fittipaldi, for your comments.
sorry. I'm so sorry. Can I now I remember there is one thing that is an equation that I didn't find correct, and I want to tell you. You can think about it. Is the
Okay.
equation 21, if I'm not wrong, on your thesis? Then, in case you will have the chance to correct. All right.
Okay.
Okay.
I would like to say that Fábio did at the beginning. Fábio did some measurements with a small range of the magnetic field, EPR measurements with a small range of the magnetic field, and we didn't see any manganese signal in the samples. It's important to say that, and maybe Fábio don't remember that. But at the beginning, we did a very small range measurements. I suppose that we don't have this manganese contribution, but at least in the considering the sensitivity that we have. Okay. Well, where is the question that Professor?
Oh.
She said that 21.
21 is there?
Yeah.
Okay.
I wrote 21. If you want, I look. Yeah.
Okay. Thank you.
Okay. Well,
Okay.
thank you so much.
Thank you too.
I definitely your contributions are important for the best document. I would like to invite Professor Christian Langhammer to make your consideration. Please, Professor Christian.
Hello, good afternoon to you. Hello, Carlos. Hello, Fabio. Thanks for the nice presentation. I would like to start with the correlation of QSM and R2 star. And you mentioned that in cortical structures, it's much worse for R2 star. Yes. So the yellow one. Could you comment what might be the origin of these low correlations here?
Of the local, okay. So I believe that lower iron concentration on these structures could be a contributing factor, but I think that from the R2 star point of view, we also have other contributions to the R2 star value. So I think that when we lower the iron contribution or the magnetic susceptibility contribution on the R2 star, other effects of R2 star are relatively higher. And that's why we do not observe a correlation to QSM, which is purely indicative of magnetic susceptibility.
And from a technical side, these cortical structures, they are challenging both ways. So QSM and R2 star is challenging to compute in the cortex. So at the very outer border of your region of interest. have you which algorithms have you used, or how did you deal with the region of interest for QSM? How did you attack this problem of cortical QSM and also ATU star?
On the cortical structures, say. Yeah. Yeah, so indeed, for the cortical structures, we have a huge problem because we have some kind of artifacts, like the background field may affect. So what we did actually is that, well, actually what I did, I eroded the brain mask before this. So of course, we kept some of the ROI outside the image because we eroded the mask. But I did this in order to improve the results here. But yes, I agree with you. We have some difficulties in the cortex regions. And I think that although I tried my best to minimize them, they are still affecting the results here. So probably that's another factor that is contributing for these differences here.
Yeah, you see, I'm a pre-Tesla person, but you have this high resolution gradient echo sequence, so the problems in the cortex are not so severe because your vocals are smaller. Okay, let me continue with neuromelanin. This is quite interesting because of Parkinson, and can you explain the ways to measure neuralin by, or to assess neuromelanin by MRI? Which methods you would use to get a clue about neuromelanin by MRI? But also for me, how could you do that with EPR? And are there other methods like histology or complementary methods?
Okay. So for MRI, I would say that we would have to study a little bit more about how to implement this, but since we did not use it in this study, I cannot say for sure the optimized parameters. But what has been observed is that the neuromelanin is visible on magnetization transfer images, especially the locus coeruleus, which is only visible by means of this contrast effect. So I have an extra image here. So yeah, from our images, we can see that the substantia nigra is visible on most images, but the locus coeruleus is absent. So yeah, our images are not suitable for locus coeruleus imaging. So what we will try to do is to apply this magnetization transfer in order to better visualize the locus coeruleus here. As for the APR and other methods, we can perform the measurements on the locus coeruleus. However, since it is a small region, a very small region, we cannot do this dividing by left and right structures, right? So because we have very few amount of local ceruleus per subject. So we have to use both sides together, and still it is very difficult, especially for APR, because the local ceruleus is noisier on APR. So one thing that on this other work that we published, what we did is that for the local ceruleus, we increased the number of acquisitions for the. So I think if I'm not mistaken, we doubled the number of acquisitions on local ceruleus in order to improve the SNR. But yeah, it is viable to do this on APR.
So and this locus ceruleus is super small.
Yeah.
And how can you see it in the tissue, and who is cutting it out, and how do you do that?
Yeah, so during the acquisition, let me present to you. No, I think it's yeah. So when we cut the sample here, we can actually see the local ceruleus here. So what we did for acquiring it is actually we did not acquired it only for one slice. So we try to acquire as much as possible. So we perform a small cut here, and then we prepare the samples in this similar way. But since it is a very small structure, before lyophilizing, before preparing them, I also use pipettes to try to remove the surrounding tissue as much as I can, so that I can only keep the pigmented region. But yeah, it's a challenging work to do.
Okay, thank you. So you assess neuromelanin and with T1 weighted or with myelinization transfer. Next question would be about decomposed QSM, but Chunlai is the expert in this, so I leave this question. But another question of interest is, some years ago we did some squid experiments, and we had small tissue samples which was then exposed to oxygen, because you cut it and it's getting exposed to oxygen. So we got another peak at 50 Kelvin. So we saw in the tissue that the oxygen of the environment is changing our measurements, and then we tried to do it in hydrogen or some argon or other non-oxygenated environment. So you mentioned you want to investigate the effect of oxygenation. How would you do that? Because at some point you have to grind this. So this is how do you plan, or how would be the idea to do that?
Yeah, it's one thing that we have to think about better. One possible way in which we can try to do this is to measure without lyophilizing the sample. The problem is that if we don't lyophilize the sample, then the ferritin signal almost vanishes. So we can't see the broad feature with non-lyophilized sample. But for the G equals 4.3, I think this would not be a problem by doing non-lyophilized sample. But yeah, for the broad feature, we would have to think a little bit more how to handle this problem.
So, but if I understood it correct, now you are taking the formalin fixed tissue, or is it fresh? Or fresh?
It's a formalin fixed.
Okay. Okay. I have a last question. It's the image processing. Forgot the page on your on the PhD, but you showed this ex vivo QSM and R2 star maps, and I'm not sure if it was in your presentation. Yeah, you showed them. In your PhD thesis, there is some face images. But how do you deal with the air bubbles? So it's tremendously a pain to have these air bubbles in the ex situ. What do you do to get images which are nice like this?
Actually, we tried to do ex vivo on 6 subjects, but out of these 6, only 2 were good enough for processing. What we did was placing the brain into the fixating media for the imaging. And what we tried to do to remove the bubbles was to move the brain into the container and gently press to try to remove. Then we also placed it on a I don't know how to say, a shaker or something that keeps shaking the container for a long time for the bubbles to come out. But what we observed is that it works on some subjects, but on others it does not work. In order to evaluate this bubble content, how much bubble, we actually did if I'm not mistaken, we did CT, computed tomography, because it's faster. So we only wanted to know if there were bubbles, and if there were bubbles, we repeated all the process. But if it does not improve anymore, then there's nothing we can do actually. But now I think that Maria found a way to use a pressure to try to remove more bubbles. So now she has an equipment there that she places the brain with the solution, and then she it's a vacuum. She creates vacuum in order to try to release more bubbles. But I don't know how good were the results yet. I did not look into the images.
Okay. Thanks a lot. Thanks a lot for being so honest. It's really have a pain getting rid of these air bubbles, and yeah, thank you for
Christian.
the question. Yeah.
But can you ask a decomposed question, please?
The decomposed question for you. No. Yeah. The question was basically if you can separate diamagnetic and paramagnetic contributions. Yes. And you're just using the paramagnetic contributions, so the image on the right. We don't have these founders, so the correlation with EPR should be much better. So would you expect it, or?
Yeah, that's what we are expecting to observe. If we are able to perform the separation of components, we expect to observe a better result of the paramagnetic component with actually both ICP-MS and APR. So we expect to have an improved result. But yeah, we still have to think about how to process the subjects that we have. I think it is feasible. Actually, it's feasible. We did it with one of our subjects, but yeah, it takes a long time. And since we did not did the ROI segmentation on one slice, but it was actually on more slices, so it will take some time to do this.
So thank you. Now I can hand over to Junlei.
Should I follow the instruction from Carlos first? So Carlos, am I ready to go?
Yes,
Okay.
please, Professor Chun-Lei.
Okay. Well, thank you, Fábio, for this very nice presentation. Maybe I just follow up on what Christian's question on decompose, but ask you a question about what the main sources are for the susceptibility in the brain. Right? So you talk on mostly on iron, but there are other chemical species that also contribute to susceptibility in the brain.
Yes.
And what are those? If you can elaborate a little bit.
Yeah. So one of these species that we believe that could have some kind of influence is the neuromelanin. So neuromelanin probably can also be affecting the magnetic susceptibility, but we have to look further into this. The deoxygenated blood seems to have a strong effect. So as we can see here, we can see lots of contribution of the deoxygenated blood. Yeah, that's one thing that we have to look. But I would say that other proteins or molecules could also be contributing on this low iron content regions, right?
Okay, so those are mostly, you're talking about mostly sources related to the paramagnetic susceptibility.
Yes.
Right, but what about the diamagnetic susceptibilities?
Okay. From the literature, what has been reported is that the main source of diamagnetism is the myelin. From what I saw from many studies, all of them assume that all the diamagnetic contribution, or most of all the diamagnetic contribution, comes from the myelin. What I believe is that, yeah, the myelin has strong contributions, but I think that there may be other molecules or aggregated proteins in the case of the disease that could be contributing to the diamagnetism as well.
But in a healthy brain, like a normal brain, you would expect the myelin to be the main contributions for the diamagnetic susceptibility. So this is kind of related to the question I've been asked earlier, and also from your presentation that you were showing the low correlation between the cortical R2 star and the QSM versus iron, for example, right? In the cortex, whether it's the entorhinal cortex or other areas, you would have relatively lower concentration of iron, but at the same time, you also have higher concentrations of myelinated axons. And so that could be one of the reasons why you will see this lower concentration, because of the R2 star value you measured, and also the QSM you measured those areas also affected significantly by the myelin. Is not dominated by iron in those areas.
Yes.
So hopefully with those kind of separations, maybe that correlation can also be improved.
Yeah, that's what we expect.
Yeah. Okay. All right. The other question is you focus very much on iron overload and talking about how iron is critical for the normal brain functions. Can you also talk about what other conditions in the brain that may be affected by the reduction of iron? Is iron overload, for example, in neurodegeneration? You talk about neurodegenerations. There's iron overload problems. I think that a couple of questions. One is iron overload a universal phenomenon in neurodegenerations, or are there other literatures also suggesting maybe there is iron reduction in some area? So that's one question. And the other question is what's the consequences of like reduced iron concentration in the brain? Are there other medical conditions that maybe is affected by the reduction of iron, not by iron overload?
Yeah, that's a good question. So about the relationship between iron overload and neurodegeneration, there are some conflicting theories in the literature because actually we don't know the relationship of cause and consequence between these. So we don't know if the iron overload is actually a consequence of neurodegeneration or if it is a cause. But from what I saw, what many studies suggest is that if we have higher iron concentration in the sample, it promotes more oxidative stress because we have more iron 2+, which can then, by means of phantom reactions, produce more reactive oxygen species, which then kills the cells and then causes neurodegeneration. But that's one hypothesis. But in order to investigate this, as I said, is if we can evaluate how the iron is distributed in the brain in the in vivo conditions. So that's a huge challenge to do. But as for the low iron concentration, its effect on the brain, as I looked, are mostly related to in the early years of the life. They are most prominent. So they seem to be related to cognitive decline during the development of infants. But if we are talking throughout the whole body, if we have low iron concentrations, it is related to also anemia. Yeah, so,
yeah, so there are those iron anemia related conditions. And there are also treatment that actually involve just iron supplements. And do you think that with the iron supplementation in a diet, for example, that is it even possible to measure the potential change in the brain or maybe in the liver that resulting from this iron supplementation using MRI techniques?
We would need to look about iron metabolism in the brain, how it is absorbed. But I believe that Yeah, I think we can see with MRI these changes.
Okay. So next question. Your data showed a lot about the kind of very interesting findings in the submission Niagara. Right? One of them is there seems to be an asymmetry, unilateral relationship between the Otsuka versus the mass spectrometry and then EPR that you observed on one side of the brain, but not on the other side of the brain. That's one thing. So maybe let me ask you this question first: one is how reliable do you think those differences you observe between the lateral differences, or is it maybe just a result of the very small sample size? Do you expect that there is some sort of lateralizations in those areas?
I think that
I mean, what is so special about the left side of the brain that shows the correlation, but the right side doesn't show?
Yeah, I think that it's mainly due to the number of subjects. I think that if we were to increase the number of subjects, we will start having some kind of symmetric results. So I think that the number of subjects are not enough to evidence this kind of relationship between QSM and RT-star on the substantia nigra. That could be because the magnetic susceptibility effect on substantia nigra is not as prominent as on the basal ganglia structures and on the right nucleus. So
yeah, my first reaction would be just small sample size. Right? I mean, it's very hard to draw a statistically significant conclusion based on two samples, but that there is a actually systematic difference between the left side of the brain versus the right side of the brain on a larger population. I mean, it's hard to say, and that may be the case. With two samples, you just have to know for sure whether that is indeed the case. Then the other thing you reported in the submission Niagara is that you suspected that because the kind of low correlations with the EPR and the mass spectrometry, that maybe ferritin is not the main source, and you are suspecting there are other contributions from neuromelanin, for example. Right?
Mm-hmm.
So from the literatures that we know, the neuromelanin doesn't contribute very much to the susceptibility. Right? For example, the study by Zina Straub, she looked at the neuromelanin in those areas, and then her results shows there is really not much contribution from neuromelanin to magnetic susceptibilities. And at the same time, there's also other data showing the iron storage in the submission Niagara areas. Maybe it is more dominated by neuromelanin rather than by ferritin. And I think with this kind of information, I wonder what kind of hypothesis or explanation you might give with regard to the non-significant correlation that you have observed.
I agree with that.
Oh, okay. Which one do you agree? So can you maybe provide a sort of explanations? Why do you see this kind of non-correlation, even though we know that actually quite a bit of iron in the submission?
Yeah. I think that firstly, this known correlation to the broad feature, well, indicates that the iron stored in the ferritin is not the main contributor to the contrast yet. So, well, in the substantia nigra, we have other iron complexes, and from what I read, they are in the neuromelanin. So the neuromelanin is also an iron chelator structure for the substantia nigra. So that's why I hypothesized that it could be contributing for it could be the source of contribution for this. But interestingly, we can't see the locus ceruleus, which also contains the neuromelanin. But I believe that's because the difference is that the locus ceruleus is actually richer in copper instead of iron. So instead of having these iron complexes in the neuromelanin structure on the locus ceruleus, we have these copper complexes in the locus ceruleus. So that's one thing that I think that could explain why these two structures have different signals and effects on the images.
So is that way from the EPR? Is that way that you can separate the iron associated with neuromelanin with the iron associated with ferritin? Is that even possible?
That's a good question. I think that it could be possible, but we would have to know how the neuromelanin signal actually is. So from what I saw from the literature, there's only one group that studied the EPR on neuromelanin, but they did it a little bit differently from what we did. So what they did is actually they prepared the sample in a way that they removed everything else from the sample, kept only the neuromelanin, and with that they observed some kind of signal. From what I remember, they also observed a signal here in the broad feature range. However, it presented a different results when varying the temperature. So I think that if we look at variable temperature EPR, we can be able to distinguish between what is ferritin and what is neuromelanin. But I think that it would require some kind of effort in order to do this, because it's not that easy.
No, I think that would be quite interesting. But probably sounds like you need to do quite a bit of calibration with different mixtures of neuromelanin and the ferritin, and then whether maybe with the temperature change that allow you to be able to separate them. I think that could be a very interesting piece of information that try to explain the iron sources and where they're located and what the relative contribution to Otsuka and accessibility within neuromelanin and within the substantia nigra area. And so I think related to this, you also talk about a very significant change in accessibility in Otsuka between the in situ and ex situ experiments, and so these changes that you saw, and I think other people have saw as well in the human brain. What do you think it is caused? Is it caused by the fixation or the fixation time, or or any other procedures during the preparation of the sample that may affect the change, and exactly what actually is changed within the brain that has caused those small differences.
So about the fixation time, I would say that it could be contributing, but to a lesser extent. But that's based on the comparison between our data to the data of a previous student. So since we observed similar results, but the fixation time between mine samples and his samples were kind of different. I don't think that the fixation time would be the main contributor for these differences, but I think that the fixation process is relevant here. So from what I saw in the literature, the explanations about this is that the fixation media can promote cross-linking of proteins, so the mobilization is affected. And for the QSM also, since the fixation media replaces the water or interstitial water, it should change the overall magnetic susceptibility of the brain. So I think that the fixation media plays an important role here for these differences that we observe.
Okay. The cross-link is going to be mostly affecting the R2 star.
Yeah.
Right, but I don't expect that the cross-link change the susceptibility of the white matter that much. And the kind of data that are available out there, people have compared the in vivo, ex vivo susceptibility differences in human brain. For example, the result of the study by Andrea Destin, they've been reported also a very significant reduction in the human brain and in the QSM. But in the mouse brain, on the other hand, actually the difference is very small. There is really no significant difference between in vivo and the ex vivo. So in your data, like the number, the value you extracted, I think in some in part is I think it's also very much influenced by the large blood clot and the veins that you will see in the in situ that you don't see in the ex situ, right? And so one thing I would recommend is when you do these comparisons, if you focus on the tissue itself, I think you will have to exclude these areas that actually it's mostly because of the blood clot or from all these very large susceptibility that you see in the veins. So, for example, if you can show the images the slides that have compare the in situ, ex situ susceptibility maps. Right? For example, over here, right, you see in situ the R2 star map and also the QSM. You see the areas are very bright, but I mean those are mostly just from the veins and also from blood clot. It's really nothing about the tissue itself, and so I think that has to be more careful when you choose the ROI, and then make sure that the vein signal are actually excluded. I mean, otherwise the ROI level measurements will be contaminated by just the fact that some of the blood are removed between in situ, ex situ, not so much about the intrinsic susceptibility changes within the tissue itself. And the other comment I want to make is from some study like an Andrea Destin study and some other studies that were shown between the in vivo ex vivo, one of the explanation what they reported based on the histological results is the fixation caused about 25%, maybe even 40% sometimes reduction of iron concentration in the tissue. Right? Somehow the fixation process caused a lot of leaking of iron out of the tissue. And so, and if that's the case, that that will actually, you were at least you were shown for your plot here as well. You see the globus pallidus in situ is very bright, right? But the ex situ one does not seem that way. So even we excluding the effect from the blood vessels, you still see in the ex situ there is a very significant reduction of the post-natal star. In the QSM as well, it's hard to see in your QSM here because you have a different scale bar. Right? On the left, you have plus minus 400. On the right, you have plus minus 200. But if you have the same scale bar, you will see the QSM and in situ is also very, very bright. So I think one of those reduction in ex situ here is really there's a loss of iron in those areas, which is kind of interesting. Why there would be whether fixation cause the reduction of iron in those areas. So that was just my comment, not a question for you. But I do have a question about the ferritin EPR spectrum. So in those experiments, the EPR spectrum of the ferritin, what's the iron loading level of the ferritin? For example, in the whole spleen ferritin, are they fully loaded, or they actually has also various degrees of loading?
Yeah, that's a good question. That's one thing that we actually are concerned about as well, because the seller they can't inform the loading factor of the ferritin that we purchased. So we don't know the loading factor of the ferritin. Yeah. So that's one thing that actually we wanted to evaluate how the loading factor affects the ferritin signal, but that would require some kind of sample preparation with the ferritin in order to produce different batches of ferritin with different loading factors. We talked to Martina about it, but at the time that I spent there, it wasn't possible to do this because we would not have time for this. But yeah, I think that it is an important thing to evaluate.
So if the ferritin has a different level of loading, what do you expect the spectrum to change? Like say compared to like a low loading versus a high loading ferritin, what would the spectrum be different? Or will it be different?
Yeah. I would expect the broad feature to change with different loading factors because the model of the broad feature is actually based on the ferriterite mineral. And the core, the most of the core is composed of these ferriterites. So what we expected is that at different loading factors, actually we have a proportional relationship between the broad feature signal and the loading factor. But we also expect to observe some differences in the G equals 4.3 signal. So with the loading factor. So because if our hypothesis is correct that the G equals 4.3 signal is actually from the iron in the shell of the protein, then we would expect it to not
So the
change.
shell, are you talking about the shell like outside the shell, or is it on the outside surface, or is the internal surface of the shell?
I would say mostly on the outside. I think.
Okay.
Oh,
Yeah, if you caught a label ion pool, I would assume there would be outside shell, right? Not the internal shell. But the broad spectrum, you don't think would change that much, or would it also change?
I think it would. The amplitude would change because the concentration would change, but the shape. Yeah, about the shape, it's a little bit harder to guess.
Okay.
But I think that from a certain range of loading factors, the shape will not be affected very much. But if we take like very low loading factors, I think that we would start to observe some changes in the shape of the spectrum.
Okay. Last question, also about ferritin. You talk about the ferritin is very stable, and then you don't expect too much change of the ion goes in and out during the sample preparation. For example, I think we talked about this before. Like say, I mean, there are certain conditions that you could actually cause the ion transport across the protein shell, the ferritin. Right? You can have ion goes in and ion goes out. Can you maybe speculate or maybe just postulate what kind of experiments, maybe EPR or some other experiments can potentially measure that kind of transport across the ferritin protein shell?
Well, that's a good question. Yeah, I think that we would need to think about it, about using EPR, because I think that the problem will be the time, right? So because EPR is not, how can I say, it's not simultaneous.
It's not very fast. There's a temporal resolution. It's not very high.
Yeah.
So this, for example, spectrum like this, how long does it take to acquire?
We obtained this with 8 minutes.
Okay.
But I think we can decrease the time if we are only looking at the broad feature. Because the broad feature is it has higher SNR, so I think we can decrease the time.
So let's say you have some sort of perturbation to the system that you can cause the ion to release from the ferritin core, and then what would you expect it to see in the spectrum, the EPR spectrum?
If we were to acquire lots of spectrum over time,
if you continuously acquire the EPR from samples, right? And then at the same time, you induce some sort of ion release from the core of the ferritin.
Yeah.
What would you expect?
I would expect that if iron is being released from the core, this broad feature would decrease, while the G equals 4.3 would increase, because then we would have less mineralized iron in the core, less ferritrides, but we have more labile iron pool.
No.
We have more.
Okay. All right. Well, that's all. Thank you very much.
Thank you.
Well, thank you very much, Professor Chun-Lei. Actually, here we are in exam, in the Fábio exam, but I would like, because it is an opportunity to discuss about the topics, I would like to comment 2 points that Professor Chunlei talk about. One is related with the loading factor. With the loading factor, I expect that the signal change, the shape of the signal change due to the zero field effect. If you remember, Fabio presented a table involving E and D values, and I expect that this value change related with the loading factor of this variety. Okay, and the other variation that I will expect is related with the behavior with the temperature. Could you move to the behavior of the temperature, please? For example, there you can see this temperature, this blocking temperature. I suspect that this blocking temperature is also related with this loading factor. Okay, and these two change, I suspect that appear when the loading factor change. The problem that we don't have control, well, we don't have control about that exactly, and we are looking for some group that can do that, just to control this loading factor and make all this and can make these measurements. Okay, and the other point that I would like to comment, even related with the comment of Christian, is related with this structure when you have a low relationship between R2 star and susceptibility values. Definitely, the reason for that is that we are not explaining the contrast of these values is not explained by the species that we are exploring here. In my opinion, that is why exactly, and because you see a very high correlation between iron concentration, for example, and R2 star, and high correlation between susceptibility value and iron concentration. But when you see with EPR, it's not clear, this relationship. So I suspect that some other species that we need to explain. Maybe some of them, maybe just melanin, maybe neuromelanin, maybe I don't know. But I think that it's important to emphasize this in the literature, to publish that, because some people in the world say, well, this tool is to quantify iron everywhere, and it's possible to assume that it's possible to quantify iron everywhere, and we suspect that it's not exactly like that. Okay. Well, actually, I am a member of this committee, but I don't vote. So I just would like to thank Fábio for all his work during these 8 years in our lab, I think. Okay, so we need to move on the private section. So I will interrupt the transmission of this section on YouTube. Okay, please. So I would like